Hello everyone, I am Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we will proceed with an explanation of all ASCP lectures from its reading list. In today's video, we will talk about hemoglobin metabolism. First, we have the hemoglobin structure. The hemoglobin molecule is a globular protein consisting of two different pairs of polypeptide chains and four heme groups, with one heme group embedded in each of the four polypeptide chains. Heme structure. Heme consists of a carbon ring, hydrogen, and nitrogen atoms called protoporphyrin IX, with a central atom of divalent ferrous iron. Each of the four heme groups is positioned in a pocket of the polypeptide chain near the surface of the hemoglobin molecule. The ferrous iron in each heme molecule reversibly combines with one oxygen molecule. When the ferrous ions are oxidized to the ferric state, they no longer can bind oxygen. Oxidized hemoglobin is also called methemoglobin. Globin structure. Each of the four globin chains consists of two identical pairs of unlike polypeptide chains 141 to 146 amino acids each. Variations in amino acid sequences give rise to different types of polypeptide chains. Each globin chain is divided into 8 helices separated by 7 non-helical segments. The helices designated A to H contain subgroup numberings for the sequence of the amino acids. Flexible non-helical segments connect the helices, as reflected by their designations, Na for the sequence between the N-terminus and the A helix. Complete hemoglobin molecule. The hemoglobin molecule can be described by its primary, secondary, tertiary, and quaternary protein structures. The primary structure refers to the amino acid sequence of the polypeptide chains. The secondary structure refers to chain arrangements in helices and non-helices. The tertiary structure refers to the arrangement of the helices into a pretzel-like configuration. Globin chains loop to form a cleft pocket for heme. Each chain contains a heme group that is suspended between the E and F helices of the polypeptide chain. The iron atom at the center of the protoporphyrin IX ring of heme is positioned between two histidine radicals. Globin chain amino acids in the cleft are hydrophobic, whereas amino acids on the outside are hydrophilic, which makes the molecule water-soluble. This arrangement also helps iron remain in its divalent ferrous form regardless of whether it is oxygenated or deoxygenated. The quaternary structure of hemoglobin, also called a tetramer, describes the complete hemoglobin molecule. The complete hemoglobin molecule is spherical, has 4 heme groups attached to 4 polypeptide chains, and may carry up to 4 molecules of oxygen. The predominant adult hemoglobin, hemoglobin A, is composed of 2 alpha-globin chains and 2 beta-globin chains. Second, we have hemoglobin biosynthesis. Heme biosynthesis. Heme biosynthesis occurs in the mitochondria and cytoplasm of bone marrow erythrocyte precursors. Heme biosynthesis begins in the mitochondria with the condensation of glycine and succinyl coenzyme A and is catalyzed by aminolevulinate synthase to form aminolevulinic acid. In the cytoplasm, aminolevulinic acid dehydratase converts aminolevulinic acid to porphobilinogen. Porphobilinogen undergoes several transformations in the cytoplasm from hydroxylmethylbilane to coproporphyrinogen 3. This pathway then continues in the mitochondria until, in the final step of production of heme, iron combines with protoporphyrin IX in the presence of ferrochelatase, also called heme synthase, to make heme. Globin biosynthesis. 6 structural genes code for 6 globin chains. The alpha and zeta globin genes are on the short arm of chromosome 16. The epsilon, gamma, delta, and beta-globin gene cluster is on the short arm of chromosome 11. The production of globin chains takes place in erythroid precursors from the pronormoblast through the circulating polychromatic erythrocyte, but not in the mature erythrocyte. Transcription of the globin genes to messenger ribonucleic acid, mRNA, occurs in the nucleus, and translation of mRNA to the globin polypeptide chain occurs on ribosomes in the cytoplasm. Transcription of the alpha-globin genes produces more mRNA than the beta-globin gene, but a less efficient translation of the alpha-globin mRNA. So the alpha and beta chains are produced in approximately equal amounts. Hemoglobin assembly. After their release from ribosomes, each globin chain binds to a heme molecule, then forms a heterodimer. The non-alpha chains have a charge difference that determines their affinity to bind to the alpha chains. The alpha chain has a positive charge and has the highest affinity for a beta chain due to its negative charge. The gamma globin chain has the next highest affinity, followed by the delta globin chain. Two heterodimers then combine to form a tetramer. This completes the hemoglobin molecule. Two alpha and two beta chains form hemoglobin A, the major hemoglobin present from 6 months of age through adulthood. Hemoglobin A2 contains two alpha and two delta chains and comprises less than 3.5% of total hemoglobin in adults. Hemoglobin F contains two alpha and two gamma chains. In healthy adults, hemoglobin F comprises 1% to 2% of total hemoglobin. Hemoglobin ontogeny. Hemoglobin composition differs with prenatal gestation time and postnatal age. Hemoglobin changes reflect the sequential activation and inactivation of the globin genes, progressing from the zeta to the alpha globin gene on chromosome 16 and from the epsilon to the delta and beta globin genes on chromosome 11. The zeta and epsilon globin chains normally appear only during the first 3 months of embryonic development. During the second and third trimesters of fetal life and at birth, hemoglobin F is the predominant hemoglobin. By 6 months of age and through adulthood, hemoglobin A is the predominant hemoglobin, with small amounts of hemoglobin A2 and hemoglobin F. Regulation of hemoglobin production. Heme regulation. Hemoglobin changes reflect the sequential activation and inactivation of the globin genes. Heme inhibits the transcription of the aminolevulinic acid synthase gene, which leads to a decrease in heme production. Heme inhibits other enzymes in the biosynthesis pathway, including aminolevulinic acid dehydrase and porphobilinogen deaminase. A negative feedback mechanism by heme or substrate inhibition by protoporphyrin IX inhibits the ferrochelatase enzyme. Increased demand for heme induces increased synthesis of aminolevulinic acid synthase. Globin regulation. Globin synthesis is highly regulated so that there is a balanced production of globin and heme. This is critical because an excess of globin chains, protoporphyrin IX, or iron can accumulate and damage the cell, reducing its lifespan. Initiation of transcription of a particular globin gene requires 1. The promoter DNA sequences immediately before the 5' or the beginning of the gene. 2. A key transcription factor called Kruppel-like factor 1, KLF1. 3. A number of other transcription factors, such as GATA1, Icaros, TAL1, P45. 4. An enhancer region of DNase 1 at the locus control region. Kruppel-like factor 1 also plays a key regulatory role in the switch from gamma chain to beta chain production. As the Kruppel-like factor 1 is an exact match for binding to the DNA promoter sequences of the beta-globin gene, it also regulates the expression of repressors of G-globin gene transcription. Globin synthesis is also regulated during translation when the mRNA coding for the globin chains associates with ribosomes to produce the polypeptide. Many protein factors are required to control the initiation, elongation, and termination steps of translation. Heme is an important regulator of globin mRNA translation at the initiation step by promoting the activation of a translation initiation factor and inactivating its repressor. When the heme level is low, the repressor accumulates and inactivates the initiation factor, thus blocking the translation of the globin mRNA. Please don't forget to subscribe to our channel, activate notifications to get our new videos. If you like our content, please press the like button, and share the video with your friends. If you have any questions, leave a comment below. Hemoglobin function. Hemoglobin has three main functions: oxygen transport, carbon dioxide transport, and nitric oxide transport. We will discuss each one of them, starting with oxygen transport. Hemoglobin's function is to bind oxygen molecules in the lung, which requires high oxygen affinity, and to efficiently unload oxygen to the tissues, which requires low oxygen affinity. The affinity of hemoglobin for oxygen relates to the partial pressure of oxygen, PO2. Low hemoglobin affinity for oxygen at low oxygen tension and high affinity for oxygen at high oxygen tension. Hemoglobin that is completely deoxygenated has little affinity for oxygen. With each oxygen molecule that is bound, there is a change in the conformation of the tetramer that progressively increases the oxygen affinity of the other heme subunits. Once one oxygen molecule binds, the remainder of the hemoglobin molecule quickly becomes fully oxygenated. With the high oxygen tension in the lungs, the affinity of hemoglobin for oxygen is high. With the relatively low oxygen tension in the tissues, the affinity of hemoglobin for oxygen is low, and hemoglobin rapidly releases oxygen. The concentration of 2,3-bisphosphoglycerate also has an effect on oxygen affinity. Lower pH and higher partial pressure of carbon dioxide in the tissues increase the release of oxygen. Hemoglobin F has an increased affinity for oxygen relative to that of hemoglobin A. High oxygen affinity of hemoglobin F provides an advantage by allowing more effective oxygen withdrawal from the maternal circulation. Hemoglobin F has a disadvantage in that it delivers oxygen less readily to tissues. Compensated by producing more RBCs to ensure adequate oxygenation of the tissues. Second, we have carbon dioxide transport. The second function of hemoglobin is the transport of carbon dioxide. In venous blood, the carbon dioxide diffuses into the red blood cells and combines with water to form carbonic acid. This reaction is facilitated by the RBC enzyme carbonic anhydrase. Carbonic acid then dissociates to release hydrogen ion and bicarbonate. The hydrogen ion from the second reaction binds oxygenated hemoglobin, and the oxygen is released from the hemoglobin. The oxygen then diffuses out of the cell into the tissues. As the concentration of the negatively charged bicarbonate increases, it diffuses across the RBC membrane into the plasma. Chloride, also negatively charged, diffuses from the plasma into the cell to maintain electroneutrality across the membrane. This is called the chloride shift. In the lungs, oxygen diffuses into the cell and binds to deoxygenated hemoglobin due to the high oxygen tension. Hydrogen ion is released from hemoglobin and combines with bicarbonate to form carbonic acid. Carbonic acid is converted to water and carbon dioxide. Carbon dioxide diffuses out of the cells and is expelled by the lungs. As more bicarbonate diffuses into the cell to produce carbonic acid, chloride diffuses back out into the plasma. Approximately 85% of the CO2 produced in the tissues is transported by hemoglobin as hydrogen ion. Hemoglobin provides a buffering effect by binding and releasing hydrogen. A small percentage of CO2 remains in the cytoplasm and the remainder binds to the globin chains as a carbamino group. Nitric oxide transport. The third function of hemoglobin is the binding, inactivation, and transport of nitric oxide. Nitric oxide is secreted by vascular endothelial cells. Causes relaxation of the vascular wall, smooth muscle, and vasodilation. Free nitric oxide has a very short half-life, but some enter the RBCs and can bind to cysteine in the B chain of hemoglobin, forming S-nitrosohemoglobin. Hemoglobin preserves and transports nitric oxide to hypoxic microvascular areas, which stimulates vasodilation and increases blood flow, hypoxic vasodilation. Hemoglobin may work with other systems in regulating local blood flow to microvascular areas by binding and inactivating nitric oxide. causing vasoconstriction, decreased blood flow when oxygen tension is high, and releasing nitric oxide, causing vasodilation and increased blood flow when oxygen tension is low. Dyshemoglobins. Dysfunctional hemoglobins that are unable to transport oxygen include methemoglobin, sulfhemoglobin, and carboxyhemoglobin. Dyshemoglobins form and may accumulate to toxic levels after exposure to certain drugs or environmental chemicals or gases. Modifies the structure of the hemoglobin molecule, preventing it from binding oxygen. Methemoglobin. Methemoglobin is formed by the reversible oxidation of heme iron to the ferric state. A small amount of methemoglobin is normally formed by the oxidation of iron during the normal oxygenation and deoxygenation of hemoglobin. Methemoglobin reduction system is NADH methemoglobin reductase pathway, normally limiting its accumulation to only 1% of total hemoglobin. Methemoglobin cannot carry oxygen because the oxidized ferric iron cannot bind it. An increase in the methemoglobin level results in decreased delivery of oxygen to the tissues. Individuals with methemoglobin levels less than 25% are asymptomatic. If the methemoglobin level increases to more than 30%, cyanosis, dyspnea, headache, vertigo, and change in mental status occur. Levels of methemoglobin greater than 50% can lead to coma and death. An increase in methemoglobin, called methemoglobinemia. Toxic methemoglobinemia occurs in normal individuals after exposure to an exogenous oxidant, such as nitrites, primaquine, dapsone, or benzocaine. If the level of methemoglobin increases to 30% or more of total hemoglobin, intravenous methylene blue is administered. The methylene blue reduces the methemoglobin ferric iron to the ferrous state through the NADPH methemoglobin reduction pathway. This involves glutathione reductase and glucose 6-phosphate dehydrogenase. In life-threatening cases, an exchange transfusion may be required. Sulfhemoglobin. Formed by the irreversible oxidation of hemoglobin by drugs, such as sulfonylamides, phenacetin, nitrites, and phenylhydrazine. Or exposure to sulfur chemicals in an industrial or environmental setting. Formed by the addition of a sulfur atom to the pyrrole ring of heme. Sulfhemoglobin is ineffective for oxygen transport. Patients with elevated levels present with cyanosis. Carboxyhemoglobin. Results from the combination of carbon monoxide with heme iron. The affinity of carbon monoxide for hemoglobin is 240 times that of oxygen. Once one molecule of carbon monoxide binds to hemoglobin, it increases its affinity and severely impairs the release of oxygen to the tissues. Carbon monoxide has been termed the silent killer because it is an odorless and colorless gas, and victims may quickly become hypoxic. Some carboxyhemoglobin is produced endogenously, but it normally comprises less than 2% of total hemoglobin. Levels may be as high as 15% in heavy smokers. Hemoglobin measurement. The cyanmethemoglobin method is the reference method for hemoglobin assay. A lysing agent frees hemoglobin from RBCs. Free hemoglobin combines with potassium ferrocyanide contained in the cyanmethemoglobin reagent. Converts the hemoglobin iron from the ferrous to the ferric state to form methemoglobin. Methemoglobin combines with potassium cyanide to form the stable pigment cyanmethemoglobin. Cyanmethemoglobin color intensity, which is proportional to hemoglobin concentration, is measured at 540 nanometer. Sodium lauryl sulfate to convert hemoglobin to sodium lauryl sulfate, methemoglobin. This method does not generate toxic wastes. Hemoglobin electrophoresis and HPLC are used to separate the different types of hemoglobins such as hemoglobin A, A2, and F. Thank you for completing the video. Remember to ask for ASCP short notes, and don't forget to subscribe. Bye.